Nobel Prize in Physiology or Medicine 2025, Scientific Background, Immune Tolerance, the Identification of Regulatory T cells and FOXP3. The immune system is a marvel of intricate checks and balances, enabling robust defenses against infections, while, in most cases, avoiding destructive responses against the body's own tissues. How is this balance maintained? This question has puzzled immunologists for more than a century. Through a combination of insightful observations and carefully designed experiments, this year's laureates of the Nobel Prize in Physiology or Medicine, Mary E. Brunkow, Fred Ramsdell, and Shimon Sakaguchi, made discoveries that provided critical answers. By defining CD4 plus CD25 plus FOXP3 plus regulatory T cells, TREG cells, and their importance in the control of self-reactive responses, their work decisively launched the field of TREG cell-mediated peripheral immune tolerance. The story is one of scientific curiosity, persistent investigations, and critical discoveries that have revolutionized the understanding of immune regulation, with relevance for self-tolerance, autoimmunity, and tumor evasion. A robust immune system is critical for our survival and health. Without it, we would be highly vulnerable, as we are constantly exposed to microbes in our environment. The immediate response following an infection is provided by the innate immune system, while the adaptive immune system requires a few days to be mobilized. A hallmark of the adaptive immune system is its ability to remember pathogens we have been exposed to previously, so we can respond quicker and more efficiently the next time we encounter the same agent. Immunological memory is mediated by our T cells and B cells, which recognize foreign structures, antigens. The recognition is mediated by antigen receptors called T cell receptors, TCRs, and B cell receptors, BCRs, membrane-bound antibodies, which are generated during lymphocyte development. Each lymphocyte has its own unique antigen receptor, and collectively, this provides our immune systems with an enormous capacity to recognize any potential foreign structure we may encounter during our lifetime. The question of how we can make so many different antigen receptors was a mystery prior to the 1980s. It was clear that there was not enough space in the genome for a one-gene, one-receptor explanation. The mechanisms that finally explained how highly diverse BCR repertoires are generated came from Susumu Tonegawa, who received the Nobel Prize in Physiology or Medicine in 1987. He demonstrated that a large set of germline-encoded genes, the so-called variable, V, diversity, D, and joining, J genes, are assembled in a combinatorial manner when B cells are formed, resulting in a unique pair of heavy VDJ and light VJ chains that make up the functional receptor in each cell. This remarkable finding was soon followed by the identification of genomic loci encoding the TCRV, D, and J genes by the groups of Tak Mak at the University of Toronto and Mark Davis at Stanford University. Like BCRs, TCRs are formed by recombination of V, D, and J genes, theoretically giving rise to more than 10 syncene distinct receptors in each individual. Most of our T cells are FOXP3 T cells, including the regulatory T cells that are central to this year's Nobel Prize. Work performed in the 1970s demonstrated that FOXP3 T cells can be divided into subsets that play complementary roles in the immune response. The two main types are CD4 plus T helper cells, which provide critical supportive functions, and CD8 plus cytotoxic T cells, which recognize and eliminate infected cells and tumor cells. Both types of T cells recognize antigens presented by major histocompatibility complex, MHC molecules. The gene clusters encoding these molecules were discovered in the 1940s and 1950s, first in mice by George Snell, and a decade later in humans by John Dawsey, earning them the 1980 Nobel Prize in Physiology or Medicine, together with Baruch Benacerraf. MHC molecules were discovered by their capacity to induce graft rejection following transplantation, but their physiological function remained an enigma. Peter Doherty and Rolf Zinkernagel demonstrated that T cells only respond to viruses in the context of MHC molecules, a finding which earned them the Nobel Prize in 1996. While the generation of vast BCR and TCR diversity maximizes our capacity to recognize foreign antigens, it comes at a cost, as some receptors inadvertently recognize self-antigens, a phenomenon Paul Ehrlich described as horror autotoxicus. It was a major challenge to unravel the molecular basis for severe autoimmune manifestations. From the work of Ehrlich and others, clinicians and researchers recognized that elucidating the mechanisms underlying immune regulation would be crucial for understanding immune-mediated diseases. So how is immune tolerance achieved and maintained? The first experiments to address this question were performed by Ray Owen, who in 1945 observed that calves with different blood groups sharing a common placental circulation developed chimerism, but did not elicit immune responses against each other's antigens. Sir Frank McFarlane Burnett predicted that tolerance must be acquired during embryonic or early life. In 1953, Peter Medawar and colleagues showed that tolerance could be induced experimentally, leading to Burnett and Medawar's Nobel Prize in 1960 for their discovery of acquired immunological tolerance. Later, Jacques Miller demonstrated that thymectomy in mice led to severe immune deficiency, identifying the thymus as crucial for T-cell development. Philippa Marak, John Kapler, Harold von Boehmer, and others provided evidence that self-reactive T-cells are eliminated in the thymus, a mechanism known as negative selection. The identification of the IR gene in the 1990s further elucidated this mechanism. R, expressed in medullary thymic epithelial cells, allows tissue-specific antigens to be presented to developing T cells, enabling deletion of self-reactive clones. This process, known as central tolerance, was long believed to be the sole mechanism preventing self-reactivity. However, some self-reactive T cells escape deletion, leading to speculation that additional peripheral mechanisms must exist. Among these mechanisms, one concept was co-stimulation, where additional molecular signals are needed to activate T cells. Mark Jenkins and Ron Schwartz showed that TCR engagement without co-stimulation led to anergy, while Craig Thompson and James Allison identified CD28 and its interactions with CD8086. Allison later received the Nobel Prize in 2018 for discoveries related to immune checkpoint inhibition. Ralph Steinman, awarded the 2011 Nobel Prize, demonstrated that targeting antigen to dendritic cells without co-stimulation could induce T-cell tolerance, introducing the concept of tolerogenic DCs. In parallel, the idea of suppressor T-cells emerged in the 1970s, but fell out of favor due to lack of molecular evidence. The field was revitalized by Shimon Sakaguchi, who in 1995 identified CD4 plus CD25 plus T cells as a distinct population essential for maintaining self-tolerance. Removal of these cells caused autoimmune disease in mice, while their reintroduction prevented it. These became known as regulatory T cells, TREGs. Sakaguchi's work defined a functional T cell subset capable of suppressing immune responses. Later, CTLA4 was identified as an additional marker, and regulatory T cells were recognized as thymus-derived. In the 1940s, a spontaneous mouse mutant known as scurfy exhibited severe autoimmune disease. Mary Brunko and Fred Ramsdell, using positional cloning, identified the mutation as a 2-base pair insertion in a previously unknown gene, which they named FOXP3. Restoring the wild-type FOXP3 gene in scurfy mice rescued them from disease. Brunkow and Ramsdell then showed that FOXP3 mutations in humans cause IPEX syndrome, a severe, often fatal autoimmune disorder. Sakaguchi's team connected FOXP3 expression to regulatory T cells, showing that introducing FOXP3 into conventional T cells converted them into TREGs. Ramsdell's and Rudensky's groups confirmed that loss of FOXP3 eliminates TREGs, causing systemic autoimmunity in mice. Together, these discoveries revealed that the absence of TREGs, controlled by a single gene, FOXP3, is sufficient to trigger catastrophic autoimmunity in both mouse and man. This understanding has profoundly influenced medicine, forming the foundation for new strategies in treating autoimmune disease, transplantation tolerance, allergy, and cancer immunotherapy. Today, over 200 clinical trials are exploring therapeutic modulation of TREGs, including polyclonal expansion, CAR-TREG engineering, and in vivo IL-2-based therapies. The story of Brunkow, Ramsdell, and Sakaguchi illustrates how curiosity-driven research and persistence can uncover fundamental biological truths and transform medicine.
Share your thoughts in the comments below. Don't forget to like this video and subscribe to our channel to show your support.